Hello and welcome back. In this video, we shall see what is called as transmission electron microscopy. So first of all, we'll see the instrumentation of transmission electron microscope. So we have a chamber which is evacuated, which is having very low vacuum, say of the order of 10 raised to power -3 to -4 Pascal, or even better than that. That means even less pressure than that. So it means it is having higher vacuum than that also. Now, first of all, we have this electron gun, which is similar to our ACM, which we have discussed in our earlier lecture. I will put the details about that in I button so that you can see how the electron gun works. Now, only the difference here is that the voltage applied to the electron gun is more, because instead of scanning, we are using here transmission. Therefore, the electron beam should transfer through a sample. So for that purpose, the energy of the electron should be higher as compared to ACM. Therefore, the voltage applied in case of TEM should be higher as compared to in case of ACM, that is scanning electron microscope. Now we have condenser lenses. Now these lenses are actually in the form of magnetic lenses. So they are electromagnets actually, or combination of electromagnets will be there. But here for representing them, we are showing it like these lenses. Okay, so this is the condenser lens. Then we have sample. Then we have objective lens, and we have the back focal plane of the objective lens. And these are the ray diagrams of these. This one is for direct beam and diffracted beam. So this gives the SAT pattern, that is the selected area diffraction pattern, and this one gives the image of the sample. Now here you can see that the electron beam is transmitted through the sample. In both these diagrams you can observe that. So therefore it is called as for transmission electron microscope. Now this is the image of actual TEM, which I had taken it from this website, from the Gel website. So this shows the actual TEM. So the whole system is in the vacuum, and then only we can observe the images here. So which is very similar to in case of ACM also. Now when the high energy electrons interact with the solid, there will be different emissions like this. Now out of these emissions, most of the time we have to take the transmitted emission here, and we have to detect them with the help of suitable detector in case of TEM. As we have seen in case of ACM, the back scattered secondary electrons and the characteristic X-rays were used. Now in this case, the transmitted electrons are generally used. So these are the transmitted electrons. Now this shows the image of SiO2 nanoparticles and the diffraction pattern also. I had taken it from these books. Now this is the crystalline diffraction pattern of a FCC steel. Then this is used for powder sample. To keep the powder sample, we have to use such kind of grid, which is 3 millimeter is there, and this type of grid we have to use for powder sample. This shows the TEM image of poliovirus. So it looks like this in the TEM image. Now this shows the defects in the steel here in this image. Now here we have some bacterium, the name exactly is given there. Now this shows the optical microscope image, this shows the ACM image, and this shows the TEM image of same type of bacteria. So you can see that how the resolution increases. Of course the colors are added actually by the software, they are not actually there. So you can just observe that how these different at different magnifications, how the magnification increases here from optical microscope to ACM to TEM. So the TEM is having best resolution there. Now for the specimen preparation, mainly we have to take care all the things which we have to do in case of ACM, that is you have to remove all the water, solvents or other materials that could vaporize in the vacuum chamber and damage the system. So we have to just remove all those things from the sample. Then the sample should be firmly mounted. Then the non-metallic samples such as some plants, etc., or they should be coated with some kind of coating actually. And then this is the different thing from the ACM, that the thickness of the sample should be small. Say if there are nanoparticles, they should be smaller than 300 nanometer. Then only we can take the TEM image of the sample. Otherwise, the electron beam will not transmit through the sample and it will be difficult to get the TEM image. Now there are various applications like in the field of cancer research, virology, material science, pollution, nanotechnology, semiconductors, paleontology and palynology. All these are the different applications of TEM. Now we have this homework here. Find the wavelength corresponding to 200 kilovolt applied voltage to the electron gun. So this is very similar to the ACM which we have done. I will put the I button link here for that detail so that you can solve this easily. And find the energy corresponding to 200 kilovolt applied voltage to the electron gun. And actually it is missing here. This question is find the resolution of the given microscope. Okay, so of course it will be a theoretical resolution because it depends upon so many other things also. So just find out the theoretical resolution here and compare TEM and ACM. So I hope that you understand this. If you have any difficulties, you can ask me in the comment section. Thank you very much.